Okay, so this is the second video, and what this is kind of all the little odds and ends that they wanted us to cover, at least the College Board wanted us to cover. When it comes to systems, they didn't actually want you to know everything about the bones or everything about the muscles. They kind of just wanted you to know, for example, how did the circulatory system evolve? Or how is it that our muscles work and how it links to our brain? So I've given Ms. Pettit these worksheets for her to hand out to you, and I want you to kind of just jot down the main points just so you get the kind of general gist of all this. And so the first thing that they wanted us to go over was actually how a muscle moves. So if I could get this to work, here we go. So you're looking at a muscle fiber, and technically a myofibril, this word that you're seeing here, is a muscle cell. It's not kind of round in shape, and it doesn't look like the crazy neurons did. They're kind of longer. And then they have these repeated bands of thick bands and then light bands, and we actually call the thin bands the actin bands, and we call the thick band the myosin, and they're alternating. And when a muscle moves, basically these thin guys move and slide along the thick ones. Now this is actually quite a complicated process. So for example, right now, hopefully, you are taking notes, and you are currently taking notes on the sliding filament theory. I'm going to say that again. You're going to want to write this down. It's the sliding filament theory. And we're seeing how these muscles move. So you can see that there's this neuron, and it is right above the muscle. And so we call this the neuromuscular junction. And when you've decided to write, basically this neuron causes an action potential to come down, and then it causes you to dump the neurotransmitter acetylcholine, which then basically causes calcium to be released in the muscle. And calcium will come to where it says troponin here, and it'll attach to the troponin, and I don't know if you can see this purple line, but the purple line rolls off, and there are little openings underneath, and so the purple line is called tropomyosin. And when it moves, so you can see this yellow here, the calcium's allowing it to move, myosin then can grab this thin filament and slide it along using energy. And that's why when we exercise, we use a lot of energy and we burn a lot of calories. So basic gist, what do you need to have one more time? It's the sliding filament theory. We are seeing thin fibers being pulled along the thick ones. The thin ones are called actin. The thick ones are called myosin. And it is a neurotransmitter acetylcholine that is causing the calcium to be released and then allowing them to bind and slide. Now here is the second concept, and this one is called counter current exchange mechanisms, and there are actually going to be three that I go over with you right now. The one we're looking at right now has to do with temperature regulation or thermoregulation. And so whether you are a dolphin or a goose, the way that their appendages work, their arteries are right next to their veins, and it's a way that they can make sure that their blood gets warmed up when returning to their body. So the blood will go down the artery, getting cooler as it goes along, and then it gets reheated up going back to the body, because everywhere along this line the artery is actually warmer than what's returning in the vein, and it heats it right back up so that the animal doesn't go back into shock. And it's kind of a cool way that they handle thermal issues. So again, it's called a counter current exchange mechanism because we've got these things lined up in parallel, and one side is causing the other side to increase in quantity. In this case, it's temperature. In this next picture, same idea, counter current exchange, but this time it has to do with oxygen. So fish, we know, use gills and we use lungs, and as water crosses the gills, everywhere along the way, water has a higher concentration of oxygen than the actual blood does in the fish's gills. So if you look down here at this picture, what they're trying to show you is that water entered, and when it entered the gills, it was at 100% saturation of oxygen. And so even though the blood had 80%, it could still give it oxygen because it was higher. And then as this water crossed, now it has less oxygen, so we're seeing 70%, but it is still higher than the 60, and that's because it has, came from this direction. So it's kind of going from right to left, and the water here is going from left to right. And so again, all along the way, it's passing oxygen. Our kidneys work in a similar way, another countercurrent exchange mechanism. So just in case you didn't know this, the kidneys are what filter your blood. So they get rid of extra salt and extra water and extra things. Even glucose, if you happen to be a diabetic, we're going to see glucose leaving. So this area here where all this redness in a circle, that's called the glomerulus, and they're a bunch of capillaries. And so the blood here is going to unload all that it can, the salts and the water, and then all along, so that's called filtration. And then after filtration, we're going to see it reabsorb, and then we're going to excrete what we don't need. But again, what we want to see here is this countercurrent exchange mechanism, where we are seeing that as the water and the salts are traveling down these tubes, they're called a nephron, and specifically this loop is called the loop of Henle, after the guy who discovered it. It's getting more and more concentrated. I don't know if you can see, it's 400, 600, 800, 1,000, which means it's getting more salty, basically, because we're seeing water leave. And then because the water left, it's actually what causes on the other side of this loop, the ascending loop, salts to leave. So it's, in this case, the countercurrent is setting up a concentration gradient where we see the water causes the salts to come out. And then later on, down here in this other tube, the salts cause the water to come out. Okay, now this image is actually an image, I'm trying to enlarge this for your sake, of the circulatory system of 3 different groups of animals. And what they want you to know in this picture actually has to do with evolution. They want you to get the idea that simple organisms like fish have a more simple circulatory system, and then they want you to get that amphibians had some adaptations and natural selection favored them to get more complicated, and so on and so forth, till we got to birds and mammals. So if you look at the fish, the first thing you should notice, it is a single loop circulation, so only one loop around. So you want to write that down, single loop circulation. The second thing you want to note, that their heart, which is over here, they only have 2 separate chambers. They have an atria and they have a ventricle. So one little place for the blood to reach and one for it to push the blood out. Now, very inefficient, because this has to reach the gills where it slows down, and then somehow we have to kind of speed that, or they do rather, they have to speed that blood back up, more gills, and then back to the heart. And so a fish, I don't know if you know this, they don't sleep, and the reason that they don't sleep is because they have to keep moving, or they're not going to get the oxygen they need to live. So that's the simplest. Now if we move up to amphibians, amphibians actually have 3 chambers to their heart, and they have a double loop circulation. So we would say evolution has favored that second loop and a little bit more division. So they have 2 places, 2 atria to receive blood, one from the body, the blood from the body, and one from the lungs. And they have 1 area here to pump it out in both directions. Better than the fish, but still not as good as ours, because good blood, meaning blood that has oxygen, and oxygen poor blood get mixed here. So it's not the best, and it's partially why amphibians like frogs and salamanders don't get very large, because they're not good at passing this oxygen along. Now, I know in this image I don't have it, but in some reptiles, that ventricle has a little bit of a division, which again, from an evolutionary point of view, would say they got, they gained that and it gave them an advantage. And then when we look at birds and we look at mammals, we have the most advanced system. We have 4 chambers to our heart, double loop circulation, and so we never mix oxygen rich and oxygen blood, poor blood, which would be the most advanced system. Now what you're looking at here is the respiratory system, and I know we're covering a lot, but like I said, they just want little bits and pieces. So for our respiratory system, I want you to think of your lungs as an upside down tree. There's this main trunk, which we call the trachea. And then we have the bronchi, which are the next, so I'm trying to find my mouse, but it's lost. The bronchi, the next branching thing, and then they branch a little bit more until we reach these little sacs that we see here on the side. They're called alveoli, and the alveoli is actually surrounded by capillaries for oxygen exchange. And so what do we need to remember here? Well, the key is surface area, surface area, surface area. There is a lot of surface area in the lungs because of the alveoli, so lots of gas exchange. What you're looking at here, again, another evolutionary concept that they want you to get is that the digestive systems of organisms have been getting more complex over time. So you're looking at a sponge, and sponges basically have every cell for themselves kind of mentality. Each cell has to digest its own food and handle its own needs, which is different than us because we actually have a stomach that does the breaking down and a small intestine that does the breaking down, and then blood takes it to all the rest of our cells. Not every cell needs to get food, which is why we can be so complex and handle so many activities. Now what you're looking at here is the jellyfish, or at least the family of cnidarians, and they have something called a gastrovascular cavity. But they only have one opening, which is still pretty inefficient. So that single opening is both mouth and anus, and so they could literally be trying to take in food and losing some food at the same time. But at least now they have a region, that cavity, that does the digesting, and the rest of the cells will benefit because they kind of come in contact with that cavity. And then finally there is what our system looks like, and we've got a track. There are two openings, food goes in one direction. We have specialized organs, all with their own little jobs to do, like the stomach breaks foods down, digest, and so does the small intestine, and then our large intestine's job is to take back water. Now what you're looking at here in this image is actually your small intestine, and again the concept of surface area, surface area, surface area. Each of those little finger-like projections are called villi, and they have little finger-like projections called microvilli. And the idea is more surface area, more absorption of nutrients and what our body needs. And in the center you can see those are our arteries and our veins, and the whole purpose is transport. And so I have covered everything. Hopefully you got everything that you needed from this. Remember that your packets are due when we get back to school on Tuesday as your extra test grade for this quarter. And we'll begin the immune system, and then that's it. We're in the home stretch. After the immune system, we begin review for our exam. Have a great break, guys. Bye.